Let me share my screen. Alrighty, can everyone see my screen? Do you want to give me a thumbs up, real or otherwise? Okay, cool. Well, I'm Kyle. I'm the host of GenSpace. I'm a microbiologist by training. My background is in microbial oceanography. As you just heard, Penny Chisholm, a researcher at MIT, and Prochlorococcus, one of the most important but little known organisms on the planet to people who aren't scientists, is what made me get my start in microbiology and oceanography. Since then, I've left the lab after my PhD, and now I'm an editor at one of the Nature journals, and I've been volunteering at GenSpace leading Planet Microbes for 1 year. This is our 1 year anniversary of Planet Microbes, which is a little wild to think about. Just over a year ago, we were like, oh, probably we shouldn't actually meet in person. So I went down to my basement and we had this meeting. And since then, we've talked about a lot of really cool stuff. And it's been the highlight of my month, every month, to get to meet with you guys and share what I think are the coolest things in environmental microbiology. So we've talked about COVID, which is not environmental microbiology, but it was on everyone's mind at that point. Hydrothermal vents and soil microbes and methane emissions and these spooky zombie fungi for the Halloween installment and going to space. We've had awesome visitors come, including someone who researches the urban microbiome using the microbes that bees pick up on their butts when they're going to collect pollen. We had last month Elise Myers, who came and talked to us about urban waterways and pollution in the Hudson. So it's been a really fun year, and I'm looking forward to the next year of Planet Microbes, hopefully in person. But we've had people come and visit us from, I think we're almost on every continent now, except for Antarctica. So we'll probably, hopefully, keep doing digital in the future as well. Beth says clapping. So that's in the plans for the future. All right, so on to this, our 12th Planet Microbes installment. And today we are talking about stromatolites, ancient microbes, and sort of a different take on viruses. Viruses have obviously been in the news a lot this past year for obvious reasons, but hopefully this paper that we read and we'll discuss today will maybe force you to change your perspectives a little bit and think about the different roles, the myriad roles that viruses can play in shaping ecology and the climate and life as we know it on Earth in ways that aren't always just negative. So the first things first, we have to introduce who we're talking about, who's the cast of characters that we're working with today, and that is stromatolites. Has anyone heard about stromatolites before reading this paper or reading the introduction to this Eventbrite? So some people have and some people haven't. We've talked about them a little bit in Planet Microbes past, I think, because I made a mistake in saying the only place on Earth where stromatolites live now, which is in Shark Bay on the western coast of Australia. I got that wrong, and Planet Microbes attendee corrected me. So this is a photo of stromatolites in Shark Bay, Australia. And they don't look like much, but they are actually living rocks that are made up of cyanobacterial mats and some friends and some foes, which we'll learn about later, and trapped bits of sediment and other little bits and calcium carbonate, and they grow up in these layered structures over thousands of years, and they are these photosynthetic, ancient, rock-like, reef-like structures that today live in very few places, most abundantly in the Shark Bay in Australia. What's cool about stromatolites is that even though they only exist in these few select locations today in limited numbers, they are some of the most ancient microbial life forms. They were some of the first organisms to ever crawl out of the primordial soup of ancient Earth billions and billions of years ago. And they still exist today, which is pretty nifty. They are also complex communities that all different microbial species that work together to create something that's greater than the sum of their parts, which we'll talk about in more detail a bit later as well. So these stromatolites are sort of like high-rise apartments, but for microbes. So if we take this stromatolite here, and then we zoom in on a cross-section of that, it's probably about less than 2 feet high, maybe. So this cross-section of it, you can see these layered structures that sort of grow up over time. And if we zoomed in even closer on the surface of this stromatolite, we would see these microscopic organisms that make it up. And these are cyanobacteria. And if we zoomed in even closer, we would see the stromatolites form these really distinct layers where different species and different organisms are layered on top of each other in these rocky domes that they have created over time. So the cyanobacteria are here at the top in green. That's their chlorophyll that enables them to photosynthesize. And then below that are different organisms, microbial organisms. And so looking at this in more of a schematic, digestible way, we can see that on the very outside, this tan layer is sand or bits of rock. Below that is the cyanobacteria. They're photosynthesizing, so they're taking in carbon dioxide, CO2, and producing sugars and spitting out oxygen as a byproduct. Then there's this oxidized layer of iron, and then 2 other photosynthetic autotrophic species of types of bacteria, the purple sulfur bacteria and the green sulfur bacteria. And they are also photosynthetic, and they're fixing carbon to grow, but they are, instead of using water, they're using sulfur compounds as their as the compound that they split apart to get electrons to do their metabolism. And so all of these organisms are working together to eat up the waste of each other and create these sort of cyclical communities that you scratch my back, I'll scratch yours, and keep things going. And then really all these stromatolites need to keep growing is a little bit of sunlight and initially water, which is in no short supply since these critters live in the ocean. And then over time, they're spitting out calcium carbonate. It's the same thing that coral reefs are made out of. And then over time, you grow really slowly and you get a stromatolite. So if this hasn't quite piqued your interest so far, I will ask you, how did stromatolites change the world? So to answer that, we'll have to think back to the very beginning of the Earth, 4.56 billion years ago, and what was the Earth like at that point in time? It was maybe not a particularly pleasant place. We didn't really have an atmosphere. It was really hot, like everything was magma because the Earth had just formed. And it was like that for slowly cooling off the, I just ruined my surprise with the advent of cyanobacteria by accidentally clicking the advance button. But for a long time, there was nothing, and the Earth was slowly cooling off, and the oceans were forming. And what was there none of on Earth at this point, really? Does anyone know? Something that we a lot today. There was water. That's a good guess, though.
Plants?
There were definitely no plants yet. Those came a couple billion years later. There was sun. You would suffocate without this.
Oxygen.
Exactly, yeah, oxygen. So for a long time on early Earth, a billion years, there was no oxygen. And that, I mean, it's totally radically different than how things are now. The first life on Earth evolved in the absence of oxygen. So that's really, compared to what we have today, a completely alien world from how it is now. And then, by some twist of evolution, these organisms that I've already shown you by accident, some twist of evolution, they cropped up. And these are photosynthetic cyanobacteria that split water, and the product of that is oxygen. And this created the Great Oxidation Event, or the Great Oxidation Crisis. So in this graph is on the x-axis or the horizontal axis, you can go from time through time, from the origin of Earth 4 billion years ago, going to the right to present. And then on the y-axis, the vertical axis, we have the percentage of oxygen in the atmosphere. So like I said before, in the very beginning, no oxygen. We only methane and other really toxic gases for today's life. And then 3.45 billion years ago, the first life evolves. And then you go for about a billion years and there's nothing. And then the first photosynthetic bacteria, by some twist of evolution, crops up. And what are these photosynthetic bacteria producing? Oxygen. And that completely changes the atmosphere of Earth, the climate of Earth, and all of the life on Earth. And I say the Great Oxidation Crisis because this advent of oxygenic photosynthesis that is splitting water and producing oxygen, that oxygen is really toxic to every single organism that evolved in the absence of oxygen when Earth was anaerobic. So these cyanobacteria actually ushered in one of the first major extinction events on Earth. Of course, because of them, they are also paving the way for the first eukaryotes, more complex organisms, the first multicellular organisms, the first organisms with a spine, enabling organisms to crawl out of that primordial soup and colonize land. And all through this time, cyanobacteria are diversifying and producing more oxygen and growing and changing the planet. So they did usher in a mass extinction, but it's thanks to them that we can be here today as humans who need oxygen to live. So Earth is, like I said, 4.6 billion years old. And for 80% of that time, stromatolites reigned supreme. So there are very few other organisms that have lasted this entire time. I mean, think of dinosaurs or trilobites or all these other animals that have gone extinct over the 4 billion, almost 5 billion years of Earth's history. In some way, stromatolites were able to eke it out and survive this whole time. And scientists still really don't have a good understanding of how they were able to grow and persist, and what happened to restrict their dominance to only a couple of places in Australia and other places around the world. But it's this incredible story of this really ancient organism that changed the world. We don't know how they did that, and we don't know how they have reached the place they are today. So that is what these scientists wrote this opinion piece about, in thinking about how maybe viruses are the missing link in what is enabling the success of stromatolites. And that is what we will talk about today. So zooming in even closer than we did before on these stromatolites, we first have to think about how they are actually formed before we can look at the specific mechanisms that enables them to do this. So in this micrograph, we've zoomed in on one chain of cyanobacteria that lives in these stromatolites. And then this sort of casing around them is a substance called exopolymeric substance. It's sort of like a sugary gel that is the foundation of biofilms, which you hear about a lot. So these cyanobacteria are secreting this biofilm, and you can see that if you zoom in even closer with this transmission electron micrograph. So this sort of honeycomb structure in the background is all exopolymeric substance, or EPS. And then on that EPS precipitates calcium carbonate or carbonate. And that's these sort of, they look like white blood cells to me, sort of, or sort of needly pom-pom-like structures. That's actually the skeleton of the stromatolite that gloms on to the EPS layer and enables them to grow bigger and bigger and bigger. And then if you repeat that again and again, 1 millimeter per year, pretty slow, over time, you will get the structures like in the image that we saw at the very beginning of the lecture. Does anyone have any questions so far about stromatolites or early Earth or?
I was just wondering, are the stromatolites in the image from Australia, are they as old as the Earth, or are they like younger colonies in
So
perspective?
that's a really good question. So those are younger questions. Those are younger stromatolites that I think are around 3,000 to 5,000 years old. And then the really, really old stromatolites, the ones that are billions of years old, those are all, we have fossil evidence of those. So you can see, actually, you should pay attention when you're walking in fancy places, because often the granite that you're walking on will be stromatolite prints on the granite. And you can, or I don't know if it's actually granite, I'm not a geologist, but you can see the sort of, they look like nestled parentheses on really fancy rocks. And those are fossilized stromatolites from billions of years ago. But the ones today, I think, are only 5,000 to 8,000 years old or younger.
Oh, and I have a question about early Earth.
Mm-hmm.
If there's no oxygen, what's fueling the fire?
What's fueling the fire? So I don't know if there was technically fire. There was a lot of magma. So it was like really hot, like molten rock. But I don't know if anything, there like wasn't a lot of stuff to be on fire. Like there was no biomass, right? There was no grass, there were no trees, there was no peat bog. There was nothing to like catch on fire. But there are other things that burn, like methane burns. So certainly, like if a meteor came through or something, or a lightning, there was lots of lightning on early Earth. So that might create an explosion, but I don't think maybe there would have been fire in the same way that like a forest fire would happen today. But there was definitely flammable gas in early Earth.
Oh, Kyle?
Yeah, hi, Frank.
Oh, hi. Sorry. Christian and I have the same question. Can these be recreated in the lab if you have the organisms that they're made out of?
Oh, that's such a good question. I have no idea if you can grow these in the lab.
You mean like sea monkeys?
Yeah, so it would be like growing a culture of sea monkeys. It would be really, it'd be slow. That would not be a project you would want in grad school. But the big challenge in a lot of these organisms is that you can't grow them in the lab. We don't know what they need to live. We don't know what nutrients they need or what partnerships they have in the environment that is really hard to recreate. So a lot of a lot of cyanobacteria in particular are really, really hard to grow. Prochlorococcus, super hard to grow. There's this one chemical mix that grows Prochlorococcus that one researcher made like 20 years ago, and they're still doling that same mix out, and it's the only one that will work. So there's a lot of like voodoo and superstition about what makes these things grow. It's really hard. But I don't actually know if you can grow the stromatolite organisms in the lab. It's a good question. Probably not, or else they would have been able to test a lot of the questions that they ask in this opinion piece in the lab for real. Like they could have isolated viruses that infect them and then actually look at how it changes calcium carbonate precipitation and stuff, which we'll talk about later. But
thank you.
Yeah, I don't think so.
There's also a related question in the chat. So
oh, sorry, I don't have I can only look at so many windows at once. So what was the chat question, Jasmine?
Yeah, just one. What are the conditions that allow stromatolites to grow in that one bay in Australia, and why aren't they common elsewhere?
Oh, that's a really good question too. So there's something about this, the like physics of this particular bay that recreates the conditions of early Earth. So the tides are really extreme in this bay. The temperature fluctuations are really dramatic, and probably the chemistry of the water, because of that, mimics as close as you can possibly get the conditions of early Earth. As for why the stromatolites don't have as wide of a spatial distribution as they used to, I have no idea. I don't think scientists know the answer to that either, but it's one of the big questions. Some scientists seem to think that the stromatolites, by virtue of spitting out all of that oxygen that paved the way for evolution to happen as it did, they sort of accidentally edged themselves out of an environment where they reigned supreme. So they paved the way for trees and multicellular organisms and eukaryotes and macroalgae and stuff. And that just, unfortunately, all those critters could grow way faster than stromatolites, which only grow at 1 millimeter per year. So yeah, there's really these microbial mats, like the cross section that I showed with the different layers. It looks sort of like cake. I don't know. It doesn't look super tasty, but it looks like cake to me, or some dense dessert. Those used to be everywhere in like the Archean Earth, but for some reason they just don't exist in very many places anymore. Only very, very, very few places do these microbial mats exist. So when scientists find one, they are like, okay, this is an analog for what early Earth conditions were like. And then they can do all of these experiments in the field to figure out how does physiology work here? How do organisms interact with each other? What's their ecology like? But yeah, they probably just edged themselves out would be my best guess.
So Kyle, are they in jeopardy? Are we in jeopardy of losing?
Yeah.
So after 4 point whatever billion years,
yeah, it's wild, right? To think that they've existed this long and that now they are, constricted to this small region. But scientists are working to protect them, and these are in protected sites. But yeah, I guess you would hope that something that existed for so long before us would exist long after we're gone, and maybe their strategy is that they're just biding their time. I don't know. Only time will tell. Oh my gosh, there's 33 things in the chat. I just figured out how to pull that up. Am I missing anything else? No. Well, if there are no more questions, and please don't hesitate, because these have been really awesome questions, every single one. So we're all about keeping the discussion going. I'm stalling here as I try and find my mouse. which tends to disappear. All right, moving on. So stromatolites, these reef-like structures, if we zoom in even closer, what you see on this, this light micrograph down here are the one potentially missing link that we haven't talked about yet and that scientists haven't really considered in the stromatolite story, and that is viruses. So this is a light micrograph where the genetic material of the organisms are stained to glow in the dark, essentially. And these big ones here are bacteria, the cyanobacteria. And then what these red arrows are pointing to, these really tiny dots, those are all viruses. So if you zoom in on this stromatolite, what you see is that, okay, we know that it's made out of cyanobacteria, a lot of cyanobacteria there, but really this thing is mostly viruses. They're just hugely abundant in this structure. And that's something that scientists haven't really considered about how these viruses are impacting the ecology and the physiology of these stromatolites. And this paper, this opinion piece goes through a couple of key features of viral ecology. And the first that I think is the most interesting as well is that viruses are the most abundant biological entity on Earth. We don't say life form because viruses actually technically aren't alive. They can't do metabolism on their own. They require a host to infect. So that's why they're biological entities. And they even outnumber all of the observable stars in the sky. And I also think that's kind of an interesting comparison between these light micrographs of viruses on the bottom and a microscope image of stars in the sky. So there's 10 to the 21 stars in the sky, and some crafty scientists have done a back of the envelope calculation to figure out how many viruses exist on Earth, and it's 10 to the 31. So it's 10 orders of magnitude more viruses on Earth, which is sort of wild to think about. The other interesting thing about viruses is that they short circuit the food web. So these light blue arrows are the normal food web that we think of. So phytoplankton are at the very bottom of the food web. They're doing photosynthesis. They're taking in CO2, producing oxygen, growing, making sugar. And these phytoplankton are consumed by slightly larger grazers. These are copepods or zooplankton. And then even larger fish, carnivores, are eating those grazers, and it goes on up the food chain. But what viruses do is they short circuit the whole thing. So they infect these phytoplankton or these bacteria. They pop them open, sort of like a balloon filled with confetti explodes, and then release all of their innards to the environment in the water. And then that prevents that biomass from moving up the food chain to carnivores and sort of recycles it back into this microbial loop down at the bottom of the food chain. So they are short-circuiting the normal path of carbon and energy and keeping it on this microbial level. So in effect, viruses are sort of popping these cells open and re-fertilizing the surrounding environment, but they're moving the direction that this food web is going. And then finally, viruses also short-circuit evolution in a way. So what are viruses? They're really just a protein coat with genetic material inside. Like I said, they can't do metabolism on their own. They are obligate intracellular parasites, so they need to infect a host. And when the virus does infect a host, it injects its DNA or RNA, its genome, inside that host, and then they make replicates of themselves and then ultimately burst out like a balloon with confetti inside. But along the way, viruses can pick up the daughter viruses can pick up errant DNA from the cells that they're infecting, or they can have mutations like we hear about with the coronavirus variants in the news. So their genetic material is changing really rapidly relative to evolution of everything else. And they can pick up those bits of DNA or mutations and then transfer them to future cells. And then those future cells could pick up those errant bits of DNA, and that's evolution happening really quickly if they can use that genetic material. So viruses are making this evolution happen on a really quick timescale. Does anyone have any questions about those 3 bits of viral ecology? Okay. And so they put all of this together, and then they start thinking about how viruses may help to build stromatolites. So you have to think about what the stromatolite environment is. It's these cyanobacteria that they show in green chains of cells and other bacteria, and then these viruses that look like little lunar landers. So the viruses are infecting these cells and they are killing them. And then just like the balloons that pop and release all of their innards, what these authors have thought about what happens in a stromatolite during viral infection is that the released innards are releasing carbonate, bicarbonate, and calcium and other nutrients. Those nutrients, just like we fertilize our plants with Miracle-Gro, are going on to fuel photosynthesis. Those organisms are then secreting that calcium carbonate even more because they are growing faster or growing more, which is seeding that stromatolite to grow. And at the same time, they could also be increasing biofilm strength through EPS production. That's that mucousy layer that we looked at a couple of slides ago. So one viral defense strategy is to release even more of this mucousy layer, and that will serve to sort of trap the viruses like they're in quicksand, and that will prevent them from reaching the cell where they'll latch on and then infect. So through producing additional EPS, they are increasing biofilm strength, collecting more bits of sand and debris, and then that's enabling those stromatolites to grow even more. So through viral infection, we have some popped cells, fallen soldiers, casualties that are sort of fueling the growth of their sister cells. Those cells might be producing more calcium carbonate, the skeleton or the backbone of stromatolites, and they might also be causing an increase in production in EPS, which is attracting those little bits of sand and other debris, further helping those stromatolites to grow. And then over time, these cells, these cyanobacteria, are becoming potentially resistant to viruses. So just like getting a vaccine, bacteria also have viral immune systems, so they can become resistant over time. And through evolutionary time, they might have, within stromatolites, selected for organisms that are better able to produce calcium carbonate, so that backbone, or better able to secrete more EPS to protect themselves from viruses. And that's like a positive feedback loop that keeps the stromatolite growing and protects them from future viral infection. So if you rinse and repeat over billions of years, then you have stromatolites. So with that, I'll ask if there are any questions for me now, or we can break into discussion groups to discuss the paper amongst ourselves. pull your faces up here. Hello.